Viruses. Further information, you can look at virus and virology. So viruses are submicroscopic infectious agents. So below the microscopes, you need more powerful than just a light microscope. Infectious agents that replicate inside living cells of organisms. Viruses infect all types of life forms from animals and plants to microorganisms, including bacteria and archaea. More than 6,000 virus species have been described in detail, and viruses are found in almost every ecosystem on Earth and are the most numerous type of biological entity. So yes, interesting setup here. And let's just take a look at this. So submicroscopic is too small to be seen even with a microscope, or a typical microscope. You need a electron microscope here. And I'm gonna do this right here. I'll just say light or optical microscope and continue further. Here is a bacteriophage. Let's see, we showed this earlier. So bacteriophages infects bacteria and archaea attached to a bacterial cell wall. So yes, supposedly that's infecting it. And again, that's just deducing from this image. And this I believe is an electron micrograph image, which involves heavy metal staining, and we'll get to that in a bit. So when infected, a host cell is forced to rapidly produce thousands of identical copies of the original virus. When not inside an infected cell or in the process of infecting a cell, viruses exist in the form of independent particles or virions. See the pronunciation here?
Virions.
Oh, virions. Consisting of the genetic material, DNA or RNA, a protein called capsid. See this?
Capsid.
A capsid, and in some cases, an outside envelope of lipids. So essentially, these so-called virus particles consist of genetic material, DNA or RNA, protein protein coating, and in some cases, an outside envelope of lipids or fats and so on. So yes, it's just a little bundle, a little ball of DNA and protein and some fat. Interesting. So the shapes of these virus particles range from simple helical to icosahedral. Icosahedral, see this one?
Icosahedral.
Or icosahedral forms to more complex structures. Most virus species have virions too small to be seen with an optical microscope, as they are 1/100th the size of most bacteria. So now we're entering near atom level size. So let me just note an icosahedron. See that again?
Icosahedron is a
icosahedron is a polyhedron. Yes, a polyhedron with 20 faces. And a polyhedron, plural polyhedra, or polyhedrons, is a 3D or three-dimensional shape with flat polygonal faces, straight edges, and sharp corners or vertices. So icosahedron is 20 of these faces. And so here's an example of a convex regular icosahedron. So it's going to be 20 faces if you count these all up. Let's go 1, 2, and you just keep counting those. And I am not going to count that. That's a regular icosahedron. And that's a polyhedron with 20 faces. Yeah, so flat polygonal faces in this case. Yeah, these are triangles there. Interesting. Straight edges and sharp corners or vertices. Interesting setup. And going further here, so it's a regular icosahedron. And just to illustrate regular versus irregular, so this figure is from tutors.com. So here's regular, has congruent sides and interior angles. So basically the interior angles are all the same here. So these are all the same setup over here. And irregular does not have congruent sides and interior angles. So these angles are all different. So the sides are different. These are different than this, and this angle is different than this, etc. Versus this one's all the same. Same, sides are same. These are all regular setups over there. And this is so here's simple, does not have self-intersecting sides. This complex has self-intersecting sides. So this one is a simple right here. Nothing's intersecting. This one's intersecting across its own sides. And over here, just more info on geometry. So concave has one interior angle greater than 180 degrees. So right here, this is greater than 180. This is greater than 180, so that's a concave shape. It's a concave shape. And here has no interior, convex is no interior angle greater than 180 degrees. So you can see the concave just means that there's a shape like this. Here there is none. Every single one's outside. This one is in like that. And this one right here, yeah, so none of these are inwards. They're all these are opposite like that. So it's convex, this is convex. This one right here is concave, this is concave over here. And this would be concave as well, because this angle right here, the interior angle here is greater than 180. So that's very fascinating. But this one's a complex one because of intersecting sides and so on. All right, continue further. All right, so here's structure of some of these so-called viruses. So as I said again, this is basically you have DNA and DNA or RNA and bundled around proteins or fats, etc. So structure of tobacco mosaic virus or TMV, and you have here RNA coiled in a helix of repeating protein subunits. You have coiled RNA like this, and you have some protein subunits. And this is supposedly a virus. All right, now let's look at this virus here. This is a transmission electron micrograph of TMV particles, which are negative stained, so the background stained instead of the specimen to enhance visibility at 160,000 times magnification. This is absurdly small. So this TMV, these are the so-called TMV or the tobacco mosaic virus. But yes, so this is what the image you get, and this is the animation that you draw up. Just quite interesting setup there. So yes, let's continue further. And again, this is negative stained. So background is stained instead of the specimen, and then you can see the contrast. We'll get to that in a bit. So structure of icosahedral adenovirus. Let's see this.
Adenovirus.
Oh, adenovirus, and you can see the shapes. There's going to be 20 sides of these. So 1, 2, 3, 4, 5, here 6. That's 6. This is 7. And then 8, 9, 10, 11, 12 there. Let's see if we can get them all. 13. That's 13, and then 14, 15. Anyways, so yes, supposedly there's 20 inside there. Let's continue further. So yes, this is the structure of a icosahedral adenovirus, or yeah, see this?
Adenovirus.
Oh, adenovirus, an electron micrograph with an illustration to show shape. So this is a supposed shape right here that looks like an icosahedral shape right here. It's supposedly like that. And this one's supposedly like that as well. And here is the the cartoon one. So the model one, and there's the images you get. So supposedly that looks like this. And yeah, to me it doesn't look too much like it. You can see there's some sort of bulges there, but again, this is a bit pushing. This is not enough evidence for my liking. So anyways, transmission electron microscopy, TME, is a microscopy. Let's go see how this is pronounced.
Microscope.
Yeah, so microscopy technique in which a beam of electrons is transmitted through a specimen to form an image. So transmitting through a specimen to form an image. An electron microscope is a microscope that uses a beam of accelerated electrons as a source of illumination. As the wavelength of an electron can be up to 100,000 times shorter than that of visible light photons, electron microscopes have a higher resolving power than light microscopes and can reveal the structure of smaller objects. A scanning transmission electron microscope has achieved better than 50 picometers right here. Yeah, so this picometer, let's type it in here. So 1 picometer, 1 pm equals to 1 trillionth of a meter. So number nanometer is a billionth, and then micrometer is a millionth. So now we're getting into the picometer level of zooming in right here of this electron microscope. Yeah, resolution there. So resolution in annular dark field imaging, or ADF. Let's see the pronunciation.
Annular dark.
Yeah, so annular dark field imaging mode, or ADF mode, and magnifications up to about 10 million times, or 10 million x, whereas most light microscopes are limited by diffraction to up to 200 nanometers resolution and useful magnification below 2000x. So this is 200 billionth of a meter. This one's 50 trillionth of a meter. And this is 10 million x this magnification. And then light is 2000 x multiplication. So yeah, a picometer is equal to 10 to the negative 12 meters, or 1 trillionth of a meter. So I just put it there. I could have just waited a bit long. So anyways, annular dark field imaging is a method of mapping samples in a scanning transmission electron microscope, or STEM. These images are formed by collecting scattered electrons with an annular dark field detector. So an annulus, see this pronunciation here?
Annulus.
So an annulus, plural annuli, or annuli.
Annuli or
So annuli or annulolus.
Or annuluses.
Or annuluses is the region between two concentric circles. So illustrate this right here. So you have, this is an annulus. So it's a region between two concentric circles. So that's this right here. There are two circles, one, and you have the second one, and it's a region in between it. So that's the annular or ring. You think of a ring over there. Yeah, so make it smaller. So you have this region between two concentric circles. Very interesting. And this concentric, I think it just means about the same axis there. So about the circling, about the same cone there. Here, just quickly pause and just check the definition. So concentric is just having a common center. And this is for circles. So there's a common center O of both of these circles. So very interesting. So let's continue further and look at this setup here. This transmission microscope. So technology. This is from technology.mate.com. So what you have is you have an electron source, and then you have the objective aperture. Or aperture is just opening there, and there's the objective lens, and it bends the light, so focuses at a specific spot, and focus probe at specimen. And then it goes through the specimen, so you're transmitting through it, and then what you end up having is you're going to form, it's going to shoot out this way around, and it's going to diffract around the object, and then you're going to form, you're going to have two setups here. You're going to have where diffracts or bends around it, and you're going to have around two circles around there is a general region, and then you'll have an ADF detector that is overlapping Bragg discs over there that overlap from different specimen going to here. This one going to here. And in the center where the bright side is, you need a bright field detector there. And there's the annular or the annulus over there. Very interesting. And now this is what it looks like. So you're going 2 million times magnification. So modern transmission electron microscope. So you're going to go get this giant thing, and you can be looking for viruses and so on. Or atoms and a lot of other stuff you can do with this powerful device. Anyways, continue further. So here's how it looks like, a diagram of it. Diagram of a transmission electron microscope. So you have an electron gun there, shoots electrons. Then you have the first condenser lens. You have first condenser lens, condenser to the point. Condenser aperture, so that opening. And then you have the second condensing lens. And then you have another condenser aperture. So you have two steps here. And then let's just zoom in more. So zoom in. And we have it over here. So the next setup is a specimen holder and airlock. And then you have objective lens and aperture. And then you have electron beam all the way here. So you get electron going here, electron gun. Now you have an electron giant beam over here. And then you have a fluorescent screen and camera. And you can detect different contrasts of colors and so on. And then you can use a computer algorithm to to parse that out and say that is which object is which, etc., or something like that. So anyways, continue further. So materials to be viewed under an electron microscope may require processing to produce a suitable sample. And the technique the technique required varies depending on the specimen and the analysis required. One technique is staining, which involves using heavy metals over here. So this is the key one. So you're staining with heavy metals such as lead, uranium, tungsten to scatter imaging electrons and thus give contrast between structures. since many, especially biological materials, are nearly transparent to electrons. So electrons just go straight through it. So what you want to do is you want to put a bunch of heavy metals on it, and then the heavy metals one that's going to be diffracting and bending or scattering the image, scattering the electrons, so you can get it a contrast in your recording medium. Yeah. So what you're actually getting with the electron microscope is heavy metals is what you're going to be recording the places where the heavy metal is scattering electrons, and since you're sticking it onto this organism, you're presumably getting a close-up view of the structure of that organism. But then that raises the question: what if you're damaging the heavy metal, especially when you're looking at very, very, very tiny particles such as viruses or bundles of DNA inside protein and lipids? Anyways, note that all images of organisms under an electron microscope are of dead organisms due to the high energy of electrons and preparation process kills the organism. Or to fix this grammatically incorrect, so which kills or kill the organisms. Yeah, so read that again. So note that all images of organisms under an electron microscope are of dead organisms due to the high energy of electrons and preparation process which kill the organisms. Yes, which is quite remarkable if you think about it. So now here's an insect coated in gold for viewing with an electron or with a scanning electron microscope. So you coat this all in gold, and then you fire a bunch of electrons on it, and then you can find the fine structures on it. But again, now you have gold plated around it, which is quite remarkable. And then when you're dealing with even like absolutely small particles like this, now you got something to think about. And now let's take a look at how absolutely small these particles are. And this figure is from visualcapitalist.com. And if you copy and paste into a Word, I believe it may compress it a bit or just harder to see. So what I'm going to do is I'm going to go into the browser itself. So if you go to the website visualcapitalist.com, you'll see that over here. So then you can click this and you see this even smaller. And let's have a look. So the relative size of particles from the COVID-19 pandemic to the U.S. West Coast wildfires. Some of the biggest threats now are also the most microscopic. A particle needs to be 10 microns or or less before it can be inhaled into your respiratory tract. But just how small are these specks? And so that's an interesting statement over there. So 10 microns, so that's micron is a millionth of a meter, and that's it needs to be that small to get into the respiratory tract. So here's a look at the relative sizes of some familiar particles. Alright, so let's take a look from the biggest to the smallest. Let's just zoom out here. So we have human hair, and for scale, so human hair for scale, so we have, this is from 50 to 180 micrometers. And this is talking about the width right here. And so, yes, it could range from bigger than, you go up to 180 and all the way to, you can get to about 50 micrometers in diameter there, right? And that's for scale. So the next one here is fine beach sand. So we can zoom in here. So fine beach sand is 90 micrometers. Remember, micrometer is millionth of a meter. So 90 millionth of a meter. And human hair is 50 to 180 millionth of a meter. And then we have grain of salt. Looks like this. It's about 60 micrometers. And then now we have a white blood cell. Over here is 25 micrometers. And then the next one is grain of pollen, which is a 15 micrometers. And so pollen can trigger, note here, this is from Harvard Health, pollen can trigger allergic reactions and hay fever, which or hay or wheat, etc., or grass, basically grass fever, which 1 in 5 Americans experience every year. Interesting. All right, continue further. So now the visibility limits for what the naked eye can see hovers around 10 to 40 micrometers. So basically around this separation point, or even over here, from here or here, depending on who's looking at it. We continue further. So the next one here is, is a dust particle. So this is PM10. Basically, this is less than 10 micrometers for this dust particle. Yeah, so these 10 micrometers, remember the note here, a particle needs to be 10 micrometers or less before it could be inhaled into your respiratory tract. So you can breathe in some dust, which is interesting. And now the next one here is red blood cells from 7 to 8 micrometers. Yeah, it's getting really small now. Now you look at respiratory droplets. This is 5 to 10 micrometers. Yeah, so now, yes, you have these 5 to 10 micrometers, so these can go in the respiratory tract. And now here's a note. Respiratory droplets have the potential to carry smaller particles with them, such as dust or coronavirus, basically liquid ball there with other possible smaller stuff. All right, let's look further. So dust particle, this one you'll call P, there's another dust particles. These are big dust particles, PM10, or it's small but not as small as these ones here. This is PM 2.5, which means this is the size here, diameter 2.5 micrometers. Now we're getting very small dust over there. Now you have bacterium, which is 1 to 3 micrometers. You get bacteria very, very small there. And then wildfire smoke, they can have 0.4 to 0.7 micrometers, so smaller than bacteria. And over here, note, wildfire smoke can persist in the air for several days and even months. Interesting. All right, the next one is coronavirus is 0.1 to 0.5 micrometers, so smaller than wildfire smoke. Absolutely tiny stuff we're talking about. All right, the next one is T4 bacteriophage. This is 0.225 micrometers. So it's this is this 0.225 micrometers is bigger than some coronavirus, but smaller than other ones there. Now, if we go even further, there's a Zika virus, 0.045 micrometers. So absolutely tiny there. Yeah, so this image is sources from this clear stream and these people over here. There's a whole bunch of people involved in this and collaborators in research plus writing. You have design, art direction. So a lot of work went into this this poster here. Anyways, let's continue further over here. So that's a figure there. Now here's another diagram. Yeah, and this figure, this is from rsscience.com. And yeah, we can look it over here. We can zoom in, or we can just go straight to the website here. And if you go here, right click, open image, and new tab, you can see the high resolution one. You can zoom in, etc. So let's look at it from here. Yeah, and just to compare the sizes, so we have 1 meter here for human, like that's like a child here, 1 meter tall. That's about 3.3 feet, etc. Something like that, or 3 feet. Anyway, so now we have in the just the widths over here. You're looking at 100 millimeters here for the heart, brain, and then liver. And then now we have the chicken eggs in between over here in the 10 millimeter range, and yeah, just a bit bigger. Now you have the fruit fly over here. The wingspans, yeah, it's below 10 millimeters over there. And number millimeters, 1,000th of a meter. Now we have a frog egg is over here. Let's zoom in here now. And then the next one here is you have a human egg, which is is smaller than 1 millimeter in size, and there is bacteria or something like that. A paramecium. And now this is conventional light microscopy, can go from 5x all the way to 100x, sometimes even 1,000x. Depending on, the more high-powered ones. So anyways, so you can go from 5x all the way 100x typically. Now you have a human egg is over there, and then now if you go below 100 micrometers, number of micrometers is a millionth of an inch, a millionth of a meter. This is a millimeter is a thousandth. So now we're going to 1,000 times smaller in the micrometer range, so millionth of a meter. And now we're looking at plant cells, so 100 millionth of a meter, and all the way to 10 micrometers. So there's a plant cell over there. There's that typical setup there. A plant cell. There's the animal cell there, and you can see the plant cell has a big void in there usually. And then next one is the below 10 micrometers red blood cells. So the human red blood cell, 8 micrometers. Now we have yeast. So this is fungi, etc. This is 4 micrometers. Now we have bacteria at the 1 micrometer range. Now let's move this over to the left. And there's mitochondria is also at the 1, and then you can see the mitochondria is inside here. And let's continue further. Now if you go to 100 nanometers, now we're going to a billionth of a meter. Yeah, in that range, now you have the so-called viruses there. And if you go 10 times smaller, you're looking at proteins themselves at 10 nanometers, and then another 10 times smaller, you're getting to the actual DNA. So 1 nanometer size there, and so the width of DNA is about 2 nanometers. Now, if you go a tenth smaller, you're looking at the atom itself, so 0.1 nanometers. And the electron microscopy goes from over here at this 10x plus all the way over here. So you're looking at 100,000 to a million plus magnification. And then X-ray crystallography can even go smaller too, so you can scan over here and get the atom size itself. Yeah, so this light microscopy, this is visible light. Now you're looking at X-ray, so a very small wavelength light, so just high frequency light over there. Fascinating stuff. All right, so now let's go further and let's look at this next image here. This is also from that same website, rsscience.com. So just another comparison. So you have the human hair, 60 micrometers, typical width over there. Now you have a red blood cell over here's 5 micrometers. So just see the comparison right next to it. So it's incredibly small. Yeah, so UM is micrometer, NM is nanometer. So it's a micrometer is a millionth of a meter, nanometer is a billionth of a meter. So now you have zoom in 10x, it looks like this. If you zoom in 10x, it's still not the width of this, or just about the width, almost. So now E. coli bacteria, this is 1 micrometers in size, and notice how small it is relative to this red blood cell. So I think they put it next to it. So this one is comparing this together. Then when you zoom in 10x, and then you compare it with a E. coli bacterium, look how much smaller it is than the red blood cell. So that's a very interesting illustration. And then if you zoom in this 5x, so it looks like that. Now you have an influenza virus is 100 nanometers, and this looks like this. This is the comparison between bacteria and so-called viruses. So it's very, very fascinating stuff. And then when you zoom in 5x, you get something that looks like this. That's very, very interesting. Let's continue further. So the origins of viruses in the evolutionary history of life are unclear. Some may have evolved from plasmids, which are pieces of DNA that can move between cells, while others may have evolved from bacteria. In evolution, viruses are an important means of horizontal gene transfer. This is this is a very interesting concept here. So viruses can transfer genes horizontally instead of using, instead of the typical methods, which is vertically using reproduction. We'll get to that in a bit. So this, which increases genetic diversity in a way analogous to sexual reproduction. Because viruses possess some, but not all characteristics of life. So you possess some, but not all is the key one right here. They have been described as organisms at the edge of life and as self-replicators. All right, so let's continue further. So MES note, horizontal gene transfer, or HGT, or lateral gene transfer, LGT, is the movement of genetic material between unicellular and or multicellular organisms other than by the, quote, vertical transmission of DNA from parent to offspring in reproduction. HGT is an important factor in the evolution of many organisms. So, for example, here is a tree of life showing vertical and horizontal gene transfers. So you have the common ancestral community set up like this, and as it goes over here, it's basically saying that there's different setups here. And let's say you had a common ancestor for all the types here, or some common ancestor for bacteria, archaea, and eukaryotes, and it goes all the way across. Or you could even have one even beyond, say something like here, and these smaller lines indicate the transmission here. So now you have a horizontal gene transfer as well as the vertical ones. So the genes of this can switch over to here, they can switch over to here, and archaea between archaea, etc. Now you have here the mitochondria can switch up from here to here, and you have plasmids involved in that gene transfer, and you have the so-called viruses that can transfer genes, etc. This is of primitive cells. Interestingly, yeah, they don't show gene transfer here of the eukaryotes after once you get to the species development setup. So that's interesting. Yeah, here, let's just draw it. Yeah, so there's none here that's showing in this image. That's an interesting concept. Let's make this maybe a bit smaller there. Okay, let's continue further. So viruses can spread in many ways. One transmission pathway is through disease-bearing organisms known as vectors. For example, viruses are often transmitted from plant to plant by insects that feed on plant sap, which is fluid transported in the xylem and phloem of plants, plant sap, such as aphids, see the pronunciation here?
Aphids.
Or aphids, which are sap-sucking insects. Yes, and viruses in animals can be carried by blood-sucking insects, such as mosquitoes. So you have the vectors that act as transporters for the transmission of this so-called virus. Right, continue further. So influenza viruses, quote, the flu, are spread by coughing and sneezing. And the norovirus and rotavirus, common causes of norovirus and rotavirus are common causes of viral gastroenteritis. Let's see the pronunciation here.
Viral gastroenteritis.
Yeah, viral gastroenteritis, which is inflammation of the gastrointestinal tract, which is the stomach and intestine, are transmitted by the fecal-oral route. See the pronunciation of this one.
The fecal.
I'll just call it fecal-oral route, passed by hand-to-mouth contact or in food or water. So the fecal-oral route, or the oral-fecal route, or the orophycal route, or the orophycal route, like that, describes a particular route of transmission of a disease wherein pathogens in fecal particles from one person pass from one person to the mouth of another. So here's a diagram. So the F diagram, the childish diagram right here. Yeah, so the F diagram: feces, fingers, flies, fields, fluids, food, showing pathways of fecal-oral disease transmission. The vertical blue line shows barriers, toilets, safe water, hygiene, and hand washing. So interesting. So you're transmitting disease. All right, that's an interesting concept altogether. Anyways, so this image is a bit unclear when I copy and paste it here. So what I'll go is go to the Wikipedia site. You can click this one, and if you click it, just to go zoom in, high definition, and click it again. I'll click this one more time. Now you get the high definition set up here. So we have feces of an infected person. So this is, I think it's they also have words here. I'm not sure which language that is. But anyways, so now you have a toilet barrier, so you could block these off. Then there's the fingers, so you're touching it, and if you don't wash. So you have flies, you have fields, and you have fluid. All right, so that could be in the water, and that is the toilet barrier, yeah, which is able to close these off. And now the next one is you have the safe water barrier. You can have it here, and then the other barrier is this one, hygiene and hand washing barrier. You could wash your hands, and it goes over to here. So all this stuff goes to the food, etc. And you're gonna have a safe water one here. And it's hygiene and hand washing barrier on both sides here. Now you eat the food, and now you have feces new host as well. All right, that's a bit childish. But anyways, continue further. So viral infections in animals provoke an immune response that usually eliminates the infecting virus. Immune responses can also be produced by vaccines. Oh yeah, this computer just blew up. Vaccines which confer an artificially acquired immunity to the specific viral infection. So let me just note, an immune response is a reaction which occurs within an organism for the purpose of defending against foreign invaders. These invaders include a wide variety of different microorganisms, including viruses, bacteria, parasites, and fungi, which could cause serious problems to the health of the host organism if not cleared from the body. Yeah, there are two distinct aspects of the immune response: the innate and the adaptive or acquired, which work together to protect against pathogens. The innate branch, the body's first reaction to an invader, is known to be a non-specific and quick response to any sort of pathogen. On the other hand, the adaptive branch is the body's immune response, which is catered against specific antigens, and thus it takes longer to activate the components involved. Interesting. And let's take a look at the vaccine setups over here. Yeah, now this is on vaccine basics, but this is in general just how do we develop immunity, and based on immunity from viruses. This doesn't talk about the vaccines yet. This is just the general theory of acquired immunity. So the body's adaptive immune system can learn to recognize new invading pathogens, such as the coronavirus SARS-CoV-2. So you have a coronavirus like this. Again, remember, it's a bundle of protein. That means it's a bundle of DNA or RNA, in this case RNA, that is enclosed inside protein and some lipids around it. So you have this so-called virus, or this bundle of genetic material, enters the body. Now you have a so-called spike protein on the surface, and this M protein in between them. You have RNA over there, and there's there's the body. Now it enters the body. Coronavirus infection. The virus uses its surface spike protein to lock onto ACE2 receptors on the surface of human cells. Once inside these cells, translate the virus's RNA to produce more virus. So human cell has these receptors here. This is called ACE2, and the spike connects to it. And just a star here is a note here. So the star means simplified. Yes, indeed. Anyways, so the virus goes into the cell through this ACE2 receptor. So virus enters the cell, and then now you have, then it creates a vesicle. So once it goes in, now you're going to have a barrier. Yeah, it's going to protrude it out. Now you're going to have a barrier inside of it, and you're going to be pretty much a vesicle with that membrane around it. And now it's enclosed like this in that vesicle. And then the genetic material gets released. So virus fuses with vesicle, and its RNA is released. So it fuses together with it as just a blob inside. And then virus fuses with vesicle, and its RNA is releases in there. And viral RNA is translated into proteins. Yeah, so somehow this gets over into the ribosomes, and then they translate it to proteins. And so virus assembly right here. Yeah, so it releases it goes over here. Now you have a new virus. It gets released. The virus released after it replicates itself. So immune response. Immune response here. Specialized antigen-presenting cells, or APCs, engulf the virus and display portions of it to activate T helper cells. And here's the diagram right here. So virus ingested by antigen-presenting cell, APC. So you have so basically antigen-presenting one. And then it grabs onto this that's released because it's supposedly a foreign material. So it grabs onto it, and then it has portions of it sticking out there. So this displays portions of it to activate T helper cells. So there's a viral peptide that's presenting it over there, and then going next here is T helper cells enable other immune responses. B cells make antibodies that can block the virus from infecting cells, as well as mark the virus for destruction. Then you have cytotoxic T cells identify and destroy virus-infected cells. So what it ends up having is, on this antigen-presenting cell, it presents these peptides, and then you have the T helper cell connects to it, and now you have setups over here. You have the cytotoxic T cell, which can recognize this and go and kill infected cells, destroy infected cells, like that. Yeah, while the B cells here make antibodies that can block the virus from infecting cells. So this B cell, the B cell makes these antivirus. So once it gets this peptide over there, it knows it. So then antivirus, anti-coronavirus antibody. So it makes these antibodies that can connect onto the virus on the surface, and then these can't infect other cells. So it prevents virus from binding or tags it for destruction. So yes, it prevents it from binding now, or you tag it now, other ones can go and destroy other cells, etc. So, and then what you have is long-lived memory B and T cells that recognize the virus can patrol the body for months or years, providing immunity. Simplify. So once they have these recognize these parts of that virus that's engulfed by this antigen-presenting cell, and then you'll have these long-lived ones, B and T cells, that live long and have recognized it. So once it sees it, it goes in and neutralizes or creates antibodies to prevent infection and so on, and also destroy other cells that are so-called infected. And this is from nature.com. So yes, it's a very it's a pretty good illustration of just the mainstream theory of this so-called this germ theory. Or not necessarily germ theory, just the idea of disease transmission. All right. So a vaccine is a biological preparation that provides active acquired immunity to a particular infectious disease. This was so active acquired. A vaccine, so infectious disease. A vaccine typically contains an agent that resembles a disease-causing microorganism and is often made from weakened or killed forms of the microbe, its toxins, or one of its surface proteins. The agent stimulates the body's immune system to recognize the agent as a threat, destroy it, and to further recognize and destroy any of the microorganisms associated with that agent that it may encounter in the future. Vaccines can be prophylactic, so to prevent or ameliorate, which is make better, the effects of a future infection by a natural or wild pathogen. It's interesting. Or therapeutic, to fight a disease that has already occurred, such as cancer. So there's vaccines for pretty much everything. So immunity is the capability of multicellular organisms to resist harmful microorganisms. So that's the immunity. A viral vector vaccine, there's different types of vaccines, is a vaccine that uses a viral vector to deliver genetic material coding for a desired antigen into the recipient's host cells. This process can be performed inside a living organism in vivo or in cell culture in vitro. Viruses have evolved specialized molecular mechanisms to efficiently transport their genomes inside the cells they infect. That's the key thing, is there's a transfer of genomes here, genetic material, whenever you speak of viruses or even vaccines. Inside the cells they infect. Delivery of genes or other genetic material by a vector is termed transduction, and the infected cells are described as transduced. Molecular biologists first harnessed this machinery in the 1970s. So viral vector vaccines use a modified version of one virus as a vector to deliver to a cell a nucleic acid coding for an antigen for another infectious agent. Yeah, so interesting. So they use viral vector, use a modified version of one virus. So you take one virus and use it as a vector to deliver the genetic material coding for an antigen for another infectious agent. So you're using another one, weakened one, or modified one to deliver another genetic material from another virus. So very interesting. So viral vector vaccines do not cause infection with either the virus used as the vector or the source of the antigen. Yeah, so this is the one that you're coding for, and this is the one you're using as a vector, so the two viruses. So supposedly does not cause infection by them. The genetic material it delivers does not integrate into a person's genome. As of April 2021, 6 viral vector vaccines have been authorized for use in humans in at least one country. So you have 4 COVID-19 vaccines and 2 Ebola vaccines. So yes, the new one's COVID-19, all of a sudden you got 4 of those. Viral vectors are tools commonly used by molecular biologists to deliver genetic material into cells. All right, let's look at the next one. So an mRNA vaccine is a type of vaccine that uses a copy of a molecule called messenger RNA or mRNA to produce an immune response. The vaccine delivers molecules of antigen encoding mRNA into immune cells, which use the designed mRNA as a blueprint to build foreign protein that would normally be produced by a pathogen such as a virus or by a cancer cell. Yes, mRNA vaccines for everything. These protein molecules stimulate an adaptive immune response that teaches the body to identify and destroy the corresponding pathogen or cancer cells. The mRNA is delivered by a co-formulation of the RNA encapsulated in lipid nanoparticles, essentially a tiny ball of fat and proteins, etc. But in this case, yeah, lipid nanoparticles, so you're looking at a tiny ball of fat, and you're putting this mRNA inside to inject. So the mRNA is delivered by co-formulation of the RNA encapsulated in lipid nanoparticles, essentially a tiny ball of fat, that protect the RNA strands and help their absorption into the cells. Among the COVID-19 vaccines are a number of RNA vaccines under development to combat the COVID-19 pandemic, and some have been approved or received or have received emergency use authorization in some countries. For example, the Pfizer-BioNTech vaccine is approved for use in adults, while the Moderna mRNA vaccine has emergency use authorization in the US. Interesting. So continue further. So DNA vaccines work by injecting genetically engineered plasmid containing the DNA. So now we're looking at DNA vaccines. Let's bold this one right here. Yeah, so you're injecting genetically engineered plasmid containing the DNA sequence encoding the antigen or antigens against which an immune response is sought. So the cells directly produce the antigen, thus causing a protective immunological response. Some cells of the immune system that recognize the proteins expressed will mount an attack against these proteins and cells expressing them. Because these cells live for a very long time, if the pathogen that normally expresses these proteins is encountered at a later time, they will be attacked instantly by the immune system. DNA vaccines have theoretical advantages over conventional vaccines, including the ability to induce a wider range of types of immune response. That's interesting. One potential advantage of DNA vaccines is that they are very easy to produce and store. In August 2021, Indian authorities gave emergency approval to this SARS-CoV-2 vaccine right here. Developed by Cadila Healthcare, see the pronunciation? Cadila Healthcare. It is the first DNA vaccine approved for humans. Yeah, just put a note here. So this one is a COVID-19 vaccine, this SARS-CoV-2 over there from India. So very interesting. And now another thing to learn up on is electroporation or electropermeabilization is a microbiology technique in which an electric field is applied to cells in order to increase the permeability of the cell membrane, allowing chemicals, drugs, electrode arrays, or DNA to be introduced into the cell, also called the and this is also called electrotransfer. So it's very interesting. And now let's take a look at this figure illustrating the differences between all these. So this is from nps.org.au, an Australian government website, or just an Australian website right here. Yeah, nps.org.au. And let's take a look at this. So conventional approaches. Yeah, so these are conventional approaches to the SARS-CoV-2 virus and the vaccine production. So you have the conventional is protein subunits. So you're just using parts of the protein of these parts right here. So you would take some of this part here, and some of these other, maybe this part right here. So protein subparts that are, these proteins are connected to these, this RNA or DNA. It's in this case RNA. So you would get those over there, and then use these and inject it into the person. And those ones are subunits, and then you get an immune response where there are, the B and T cells can recognize these for future infection. And then you can also have this virus-like particles. You can have this setup here just with the spike, but nothing else, no genetic material inside, just that. And then that you can also use whole virus vaccines. And this is an attenuated or weakened virus, and this is an inactivated virus. So it's attenuated, so you can make it weaker by removing this or switching this up so that it doesn't do the bad stuff, or it doesn't replicate, etc. Yeah, or this inactivated virus right here. Yeah, this inactivated could be set up like you could break it apart and etc. So it's pretty much the same thing, but the genetic materials modified. We'll get to the attenuated. This one's weakened. This one I think will just be not replicating. Anyways, continue further. And now you will have is novel approaches. You have these two setups here. So you have the nucleic acid vaccines. So you have the DNA vaccine is just the plasmid that you're throwing in there. Now you have the mRNA vaccine. You have that ball of fat with RNA inside. Then you inject it into the person. And this RNA is to code for specific antigens, such as these so-called spike proteins. And this one too could be coded for the spike protein, or this as well in the RNA side. Other one is the DNA side. Remember the genetic transfer where you're transferring via plasmids, etc. If you go all the way back to here, this one's an interesting illustration here. So you can have horizontal transfer. You get the plasmids transferring from different organisms to other types as well. And then you have mitochondria. You could also have virus transmission as well, as it illustrated here. Yeah, so in evolution, viruses are an important means of horizontal gene transfer, which increases genetic diversity in a way similar to sexual reproduction. So very, very interesting. So keep that in mind. Yeah, so when we're dealing with viruses, vaccines, especially DNA and RNA vaccines, you're dealing with genetic material transfer. Yeah, it's very interesting. And now the next one is a viral vector vaccine. So you have a non-replicating viral vector. You have something that looks like this, and so it's a different virus right here. Yeah, so it's a different virus that's containing the genetic material to encode for specifically this one here. And now this one, remember, so these ones are different colors just to illustrate that this is a different virus holding it. So this is a replicating viral vector that, you can replicate. This one doesn't replicate. And they both encode for that specific part of this one here. But they use different viruses there. So viral as the vector. So you use different viruses as the vector. So this is from April 2020, just to keep that in mind, because it could be more updated types there. And now let's look at further on this protein-based one. This is the protein-based one. There's several types over here. These are conventional approaches, and the protein one is protein subunits. All right, so we have this. We could even zoom in. Let's just zoom in a bit. So protein-based vaccines. So you have protein subunits is one type. So you have 28 teams are working on vaccines with viral protein subunits. Most are focusing on the virus's spike protein or a key part of it called the receptor binding domain. Similar vaccines against the SARS virus protected monkeys against infection but haven't been tested in people. To work, these vaccines might require adjuvants, which are immune-stimulated molecules delivered alongside the vaccine. So that's interesting. So you have another immune-stimulating, but they're not part of the vaccine, or not part of the vaccine. But you're injecting it with the vaccine as well. I mean, they're not part of the so-called genetic material or the virus material that you want to develop the specific acquired immunity for. But this is just to stimulate the immune system. So as well as multiple doses. To work this might require adjuvants or as well as multiple doses. All right, so now we have this M protein, there's a spike protein, and just a note here, so most are focusing on the virus's spike protein or a key part of it called the receptor binding domain. So that one, I believe that one they're talking about this part right here. Now you have the spike protein on this end over there. It's interesting. Yeah, and here I just teleported higher up. So you see the spike protein on top there. There's an M protein at the bottom there. So it's very interesting. And continue to scroll back down. This one here has even more setup here. So it has extra stuff on top there. So you could use some of that as well. But anyways, we're going to go over here. So the spike protein, M protein. And then you put that in the vaccine. So you inject into the body. And then what happens is these particles, the antigen presenting cell, or the APC, it finds these and then now it binds parts of those onto here. So coronavirus peptide, and then there's an immune response, and they recognize it. T and B cells recognize this over here and then develop immunity from that or memory from it. And then you can also use virus-like particles, which is still part of the protein-based vaccines. Because you can, so empty virus shells mimic the coronavirus structure, but aren't infectious because they lack genetic material. So 5 teams are working on virus-like particles, VLP vaccines, which can trigger a strong immune response, but can be difficult to manufacture. And let's go to the bottom here. Actually, no, this is all from here. This is all from this figure. I believe it's from here. And this is on, COVID-19 vaccines and the research from April 2020. All right, continue further. All right, yeah, so this is, so 5 teams are working on this, or we're working on it, which can trigger a strong immune response, but can be difficult to manufacture. Because you got to manufacture, just an empty shell to mimic that virus. And that's over, it could be, right here, virus-like particles there. That's on that part. Continue further. And yeah, so you injected that in, this goes in, and then the peptide, coronavirus peptide goes over here, and there's an immune response. So notice there's always this setup over here with this antigen presenting cell there. Very interesting. All right, so now let's continue even further. And now let's look at the virus vaccines. So we had, this was the protein one here. And now we're going to be the whole virus vaccines. This is the protein subunits, virus-like particles. Now you actually have the whole virus one, and you're using, this is the whole virus one actually. Where you have weakened one or inactivated one over there. So let's go back here. So virus vaccines. So weakened or attenuated. So a virus is conventionally weakened for a vaccine by being passed through animal or human cells until it picks up mutations that make it less able to cause disease. And this is Codagenics in Farmingdale, New York, is working with the Serum Institute of India, a vaccine manufacturer in Pune, to weaken SARS-CoV-2 by altering its genetic code so that the viral proteins are produced less efficiently. So it's weakened, so it passes through animals constantly until it weakens via mutation. So very, very interesting. So and then you put that in the vaccine, and you can do the same setup here. But then here's inactivated virus. So in these vaccines, the virus is rendered uninfectious using chemicals such as formaldehyde or heat, making them, however, require starting with large quantities of infectious virus. You got to get a whole bunch of them, and then you're going to weaken them, etc., with heat or chemicals, and so on. And then the same thing, you insert it into the cell, into the body, and then you have these antigen-presenting cells go in and absorb that, engulf it, and then it gets the peptides or parts of it and presents it for the immune response system. So there's coronavirus peptide and so on. So the setup is always the same there. So now let's look at viral vector vaccines, and that's over here in the diagram is viral vector on this setup over here. And then the next one is nucleic acid vaccines after that. This is viral vector one, and these are the novel one, new types. And this one uses other virus as vectors for coding for that specific virus you want. Let's go back here. Alright, so viral vector vaccines. Remember, you're using other viruses as vectors to deliver genetic material of the target virus. So replicating viral vectors such as a weakened measles virus, you use that as the vector. So the newly approved Ebola vaccine is an example of a viral vector vaccine that replicates within cells. So such vaccines tend to be safe and provoke a strong immune response. However, existing immunity to the vector, not the target virus, could blunt the vaccine's effectiveness, however. That's interesting. So you'll have a weakened measles virus like here, and there's a viral gene, etc. What you're going to do is you modify it so that the spike that it produces is actually the coronavirus spike, and this one can replicate and so on. So it's the target one that you want to build an immune system for the spike ones to recognize the coronavirus. But however, so this one, if people already have immune response to the viral vector, then this might get destroyed quickly, and this won't be as effective of getting this into the immune system. And let's just erase all this. And now let's look at the non-replicating one over here. So non-replicating viral vector, such as adenovirus. So there's currently no licensed vaccines use this method. So yeah, you'll use this adenovirus as a viral vector and it's non-replicating. But they have a long history in gene therapy. So those are modifying the genes if you want to treat for cancer, etc. Booster shots can be needed to induce long-lasting immunity. That was just pretty interesting. So booster shots, subscription-based vaccine induction. So US-based drug giant Johnson & Johnson is working on this approach. So what you'll have is a coronavirus spike over here. Again, you want to code for that. There's viral genes somewhere inactive, so to prevent replication. And then you'll have the vector right here, such as adenovirus. And this one here is weakened measles. Yes, and in both of these cases, what you're going to have is when you inject it in, and again, you have that, so virus replicates in this case, but in this one, it wouldn't replicate. And then what you'll have is, the antigen presenting cell engulfs these, and then it puts the coronavirus peptides, or yeah, in this case, the spike peptides, a specific one that you want in to be recognized. So it'll put some of those antigens, some of those peptides on here, and then you get the immune response and so on. So that presents it to the T and B cells. They go and look at it and memorize it for a future. All right, so now let's look at the nucleic acid vaccine. So now you have the two types of setups right here. So you have the DNA vaccine and the RNA vaccine. And this is why I went over the electroporation, just to make it easier for the cell to go into, for the vaccine to go into the cell. So what you're going to have is you're going to have a coronavirus spike gene. This one's just a plasmid with that specific spike gene that you want to be recognized. And you put that inside over here. So that's a DNA plasmid, and there's the electroporation there. So you're going to put this in a vaccine. And then what you're going to do in this case, you're going to insert it in, and usually including a process called electroporation, which creates pores in membranes to increase uptake of DNA into cell. Because you're basically putting a naked plasmid inside the body, and then you want to weaken the cells around using just an electric field to break it apart, so that this can go all the way inside. Now you have a plasmid going to the nucleus, and then what happens is once it gets into the nucleus, it will translate into mRNA or transcribe into mRNA, and then this goes in ribosomes and translates into that specific spike protein setup over there, and then this goes into that antigen presenting cell for the immune response. So again, that's the same cycle over there. All right, so now the next setup is RNA vaccine. So basically you're going to have a ball of fat with the RNA inside. So you have RNA is enclosed in a lipid coat so it can enter cells. And then there's the RNA with the specific spike protein gene over there. And then what you can do is throw it in there, or spike protein sequence that's transcribed from the DNA already. And so you're going to put that in there. And then you're going to inject it, and this goes in because it has that lipid layer. It can go in here, connects to the cell, and then they can deliver this inside. And then this goes to the ribosome and translates it into proteins. So viral proteins, etc. goes over here, and there's a coronavirus spike peptide on the antigen-presenting cell that engulfs these. And then again, you get an immune response. Again, this is from April 2020. So if RNA and DNA-based vaccines are safe and easy to develop. To produce them involves making genetic material only, not the virus. But they are unproven, no licensed vaccines use this technology. This is from April 2020, but the RNA one does, and I believe the DNA one has been. Yeah, the DNA also has been approved in India, and the RNA is approved in the US and so on. And the keyword here is it's easy to develop. So right now this is from April 2020. So this is unproven, no licensed vaccines, but they are licensed now in some parts of the world. So the keyword is easy to develop. So expect a whole bunch of these vaccines and booster shots, etc., to be on the way. Because if it's easy to develop, that's going to mean more sales. For big vaccine manufacturers.